More exam technique points from the November 2011 paper. This question is asking you to identify the molecular formula of the two disaccharides. So, points to remember. All the monosaccharides mentioned are hexose sugars, therefore their molecular formula is going to be the same. So to calculate what it is you need to add them together and then take away the water molecule that is lost in the formation of the glycosidic bond. Okay, so you've got C6H12O6 plus C6H12O6. Okay, so that is going to be C12H24O12. However, it's not as straightforward as that because you then need to take away the water molecule that was lost in the formation of the glycosidic bonds. So then we need to take away 2 H and 1 O. So that's going to be C12H22O11. And it's going to be the same for both disaccharides. So you then need to look along and look for C12H22O11 in both columns, which is here. So in this question, you're being asked to identify what the lines do. Because it's a relatively simple graph, the easiest thing is just to identify what X, Y, and Z do, and then to find them on the table. So, to identify them, X here is showing a normal uncatalyzed reaction, Y is showing a catalyzed reaction. You know that because of the difference in activation energy, and Z is showing the energy change in that reaction, and then it's just a simple case of following along the table until you find the thing which has all of those items, and the answer will give you there is D. You often get definitions questions in multiple choice papers, and the trick is to read each definition carefully and decide whether it is true or not. Once you've found the correct one, do check the others just in case. So, which statement defines active transport? The movement of large molecules through the cell surface membrane? No. The movement of molecules of ions from where they're in low concentration to where they're in high concentration? Yes. Let's just check the others. Movement of ions from high to low? No. Movement of water molecules? No. So the answer is definitely B there. And then the next one, single-celled animals that live in fresh water have a vacuole that contract regularly to remove excess water. Single-cell plants that live in fresh water do not have a similar vacuole. Okay, so that is obviously there to protect the animal cell from getting too large and the cell membrane rupturing. So plant cell cytoplasm and animal cell cytoplasm both have lower water potential than fresh water. Well, that's true, but it doesn't explain this phenomenon, so no. Plant cell sap has the same water potential as fresh water. Animal cytoplasm has a lower water potential than fresh water. Nope. Plant cells are impermeable to water. Nope. And so this last one, plant cell walls restrict the entry of water. Animal cell membranes allow the free entry of water. Yes, please. So the answer is D. Another one of your favourite graph questions. In this one, you're being asked to calculate the total time that the atrioventricular valves and the semilunar valves are both closed at the same time. So your first thing to do is to identify where the atrioventricular valve closes. So the atrioventricular valve closes when the pressure in the ventricle is greater than the pressure in the atrium. So here, at 1. Now the semilunar valve is the one between the ventricle and the aorta. So the semilunar valve is closed all the time that the pressure in the aorta is higher than the pressure in the ventricle. So it's closed all the way up here, and it's also closed between 3 and 4. Now 4 is the point at which the AV valve opens again. So the total time is going to be the time between 1 and 2 and between 3 and 4. So you then need to read along the graph. So 1 is at 0.12 and 2 is at 0.15. So that's 0.3 seconds. And then 3 is at 0.36 and 4 is at 0.4. So that's 0.4 seconds. So that's a total of 0.7 seconds. Sorry, 0.0. All of those. Oh dear. Bad maths. So C. This is a good example of the elimination technique. So you look at this and you see all of these things that you have to identify, and your brain goes slightly into a spin. But actually, if you look down each column, what you want to do is you want to look for a column where all 4 options are different, which is this column here. So each thing in column 1 is different. So if you can identify what number 1 is, then you have answered the question. Now number 1 is an enzyme. It is the enzyme involved in converting carbon dioxide and water to number 2. That enzyme, the only enzyme in this list, is carbonic anhydrase. Bob's your uncle. B is your answer. In this question, it's simply asking you where is the highest concentration of sucrose, A, B, C or D. Well, it's fairly straightforward logic. Sucrose has to move from here to here and then down to here and then into there. Okay, this movement from B to C wouldn't happen if the concentration of C was higher than the concentration of B, so we can cross that off. Likewise, this movement, C to D, is a relatively passive process, so that's not going to be higher. You have an accumulation of the sucrose in B. Remember, it's a double stage jump from here to here. So actually, the concentration in B is going to be higher than the concentration of A. So there's your answer, B.